Good afternoon. I'm John Lors, Director of the National Institute of General Medical Sciences, and I want to welcome everyone to the DeWitt Stetton Jr. lecture today. I want to start by telling you about Dr. Stetton and some of his background. Dr. Stetton spent most of his career at the NIH, serving in 2 institutes as well as the Office of the Director, but he was probably best known as the NIH Deputy Director for Science, although one of his other great achievements was as serving as the director of NIGMS. In recognition of his many contributions within and outside of NIH, Dr. Stetton had the rare distinction of not only having a lecture named after him, this lecture, but also a museum, the DeWitt Stetton Jr. Museum of Medical Research. Now to our speaker today, Dr.Sengupta joins a very distinguished list of Stetton lecturers. She's made major contributions to understanding the molecular and neural basis of sensory behavior in C. elegans. Her research combines multiple approaches to tease out the complex interplay between an organism and its environment. Piali began this work as a postdoc at UCSF with Corey Bargman, where she identified odor 10, an olfactory mutant, and showed that the gene encodes a receptor that's required for the behavioral response to a specific odor. This finding not only advanced understanding of the molecular basis of odor sensation, but it also provided a tool for uncovering the relationship between genes, molecular events, and behaviors. Since then, Piali has systematically identified many of the genes, neurons, and molecular pathways involved in sensing chemical and thermal cues and translating them into specific changes in behavior and development. One of her focus areas is primary cilia, which were first described in 1898, but considered by many to be vestigial, until that is fairly recently. But they are not vestigial, and Piali's work is illuminating how these structures form and function, the genes involved, and what happens when they're defective. I'm very pleased to say that NIGMS has supported Piali's research since he joined the faculty at Brandeis in 1996. Please join me in welcoming Piali.
Well, thank you very much for that kind introduction. It is a great honor and privilege to be invited to present a talk named after Dr. Stetton. So while preparing for this talk, I read a biography of Dr. Stetton that was published by the National Academy, and I learned that near the end of his life, he suffered from a retinal degeneration and blindness. And as it turns out, dysfunction of the sensory organelle that I'm going to be talking about today, the primary cilium, actually underlies many forms of retinal degeneration, including the form that Dr. Stetton suffered from. And as you'll see during the course of my talk today, there has been an absolute explosion of information and knowledge in the field of cilia biology in the years since his passing in 1990. So before I get into that, though, I wanted to spend a minute just talking about the role that NIH and especially NIGMS has played in the establishment and progress of my research program. So as John mentioned, I started my lab in 1996 at Brandeis, and not knowing any better, submitted an R1 almost immediately. And so this was during the times when NIH was very well funded, and I got the grant. And so this is the NGA for my very first award, which I very carefully saved. So this is in 1997, and I'd like you to note the name of the program officer, Mike Sesma. So fast forward to 2015, and I've been very lucky that this particular grant has been funded continuously since then. And this is the NGA for my latest renewal in 2015. And notice that it is again Michael Sesma, 19 years later. He did take a break there in the middle, though, to go do something else a little more interesting than manage my grant. And I had another wonderful program officer, Lori Tompkins, who I understand is currently enjoying her retirement in some state called Hawaii. So the support and advice that I received from these program officers has really been invaluable in my career. And I want to take this opportunity today to thank them and also GM for their interest in and support of basic research, especially in model organisms, even during these trying times. So in addition to receiving funding from GM, our work has also been funded by NS, NINDS, and NIDCD both in the form of research grants, but more importantly, in the form of fellowships for my grad students and postdocs. And I again wanted to thank the NIH for their continued and hopefully continuing support of our work. So on to science. So the question that I've been interested in since my grad school days is really understanding how animals sense the complex world around them and then translate this information into very specific changes in behavior and development. So take, for instance, this picture of a very greasy breakfast frying in a pan. So if this was in real life, you'd be receiving a lot of different types of stimuli from this. You would smell the bacon frying, you could hear the sausage sputtering, you could watch the egg hardening, you could feel the heat from the fire. And all of these stimuli together would go into your decision of what you were going to do with this breakfast. But in addition to these stimuli, there are a number of other factors that would also play into your decision. These include your internal state. So for example, have you eaten recently? Are you full? Your physiological conditions, so for instance, perhaps you're tired, perhaps you're sick, and also, of course, your past experience. Maybe you had a very bad reaction to eating eggs before. And so the overall question that my lab is interested in is really understanding how animals sense and perceive these sensory stimuli and then translate this information in the context of internal state, past experience, and external context to drive very specific behaviors and developmental decisions. So as John mentioned, over the years, my lab has done a lot of work on the molecular neural basis of both chemo and thermosensation, mechanisms by which animals respond to chemical and thermal cues in the environment. But today, I'm going to be only talking about some of the work we've been doing on the cellular basis of sensory signaling. So it's been known for a long time that sensory signaling molecules, signal transduction molecules, such as those in olfaction or phototransduction, are localized to these specialized structures called cilia. So in the case of olfactory neurons and the olfactory epithelium, molecules such as these odor and G protein coupled receptors, adenyl cyclase, G proteins, are all concentrated in this bunch of cilia that are the end of dendrites. Same thing happens in the retina. So there are molecules such as the rhodopsins, conopsins, transducins, cyclic nucleotide gated channels are all concentrated in this specialized ciliary region referred to as the outer segment that's found in both rods and cones. And it's been known for a long time that defects in the ciliary localization or trafficking of these proteins or defects in the structure of these cilia lead to not only defects in sensory signal transduction, but can also actually lead to neural degeneration and ultimately cell death. And so what that would mean in the case of the olfactory neurons is, of course, anosmia. You wouldn't be able to sense any chemicals around you. And in the case of the retina, this would lead to blindness. So it has now become abundantly clear that cilia are not a characteristic of just specialized sensory neurons. And in fact, they're ubiquitous organelles. And they come in two sort of general broad flavors. So there are the motile cilia, also called flagella. So for instance, those present on this blue-green algae Chlamydomonas, which helps this organism to swim, they actually also have sensory functions. And in humans, motile cilia are found on a subset of different tissues. This picture shows you the cilia that are present on the epithelia lining your airway, and whose beating actually is really important for mucociliary clearance. So today, I'm not going to be talking about this category of cilia at all. What I'm going to concentrate on are these non-motile cilia, which are also called primary cilia. And as it has now turned out, these primary cilia are actually present on nearly every single cell and tissue type in a metazoan. So what I'm showing you here in red is a little cilium that's sticking out from the soma of a hypothalamic neuron in the brain. And this is a cilium with a bulbous ending present on a kidney cell. And so these cilia stick out into the environment, and sometimes they've been referred to as cellular antennae. And they respond to external cues and transduce them into the cell. And so given their critical roles in maintaining cellular and organismal homeostasis, it is therefore not surprising then that cilia dysfunction has now been implicated in a huge number of heterogeneous disorders, collectively termed ciliopathies. And so this lists a few of them here. There are tens more. And these are all characterized by a number of different symptoms that are reflective of cilia dysfunction in different tissue types. So for instance, many of these ciliopathies result in polycystic kidney disease. So you get these fluid-filled cysts in the kidney. And this results from defects in renal cilia function. Obesity can arise from defects in the formation, the structure or the function of cilia present on neurons in the hypothalamus, the appetite control center of the brain. And then polydactyly can actually arise from defects in sonic hedgehog signaling during early development. And as it has been shown initially by Catherine Anderson's lab, sonic hedgehog signaling actually requires primary cilia. And so it's become really important to try and understand how these structures form and how they function. So given our interest in sensory signaling, there's one aspect of cilia that particularly interests us. So most of the cilia found on different tissue types actually have a very simple rod-like structure, like this kidney cilium I'm showing you here. But that's actually not the case for most of these sensory cilia. And I already showed you that in cartoon form before. But in the case of the olfactory neurons, for example, you have this sort of bunch of cilia that emanate from the dendritic knob. And so while these cilia are usually somewhere between 5 to 10 microns or so, some of these cilia can be as long as 100 microns or longer. In the hair cells of the inner ear, you have these kinocilium, which plays a major role in orienting the hair cell bundle. And these structures in blue are actually, unfortunately, named stereocilia. They're actually not cilia structures. They're actin-based. And then, of course, as I showed you in the case of photoreceptors, you have these outer segments of the rods and the cones. And what you can see very obviously here is these structures are quite diverse in their morphologies and very different from this simple rod-like structure present in most cell types. And so we know today a lot about how cilia are formed and how they function. What we don't quite understand still is how these different types of cilia come to be, and more importantly, how these different morphologies contribute to the special functions of these sensory neuron types. And that's going to be the theme that's going to run through my talk today. So what are cilia? Well, cilia are these microtubule-based organelles. They're all templated from the basal body, which derives from the mother centriole. They have a microtubule-based structure referred to as an axoneme, and this is surrounded by a specialized plasma membrane, which houses those signaling molecules I referred to before. So one characteristic feature of cilia is that there's no protein translation within the cilium. So everything that's required for building or the functioning of the cilium must be trafficked to the base of the cilium and then trafficked within the cilium. And so this process of transport within the cilium is an incredibly conserved process, all the way from chlamydomonas to humans, and it's a process referred to as intraflagellar transport, or IFT. And this works in the anterograde direction via the plus-ended motors kinesin, and in the retrograde direction via dyneins. And I'll have more to say about IFT a little later on. The other characteristic structure of these cilia is this complex of proteins at the cilia base, which we call the transition zone. And essentially, you can think of the transition zone as a ciliary gate. And what that does is that it maintains the protein composition of the cilium by keeping ciliary proteins in and keeping non-ciliary proteins out. So we know a lot about the structure. We know a lot about the trafficking mechanisms that bring proteins to the base and traffic them within the base. And I think at this point, it might be useful to actually take a step back and look at the history of cilia biology, because I think it provides some very interesting lessons in the process of scientific inquiry. So the first motile cilia or flagella were noticed by the famous Leeuwenhoek, and he noticed these on protozoa. Following this, however, the immotile cilia, although Zimmerman gets the credit for this, as John pointed out, they were actually first noticed earlier by the famous Langerhans, the same pancreatic Langerhans, and Kowalewski. And this is actually, they noticed this in the marine chordate Amphioxus. And this is a figure from one of these papers where you can see these, what they refer to as these hair-like structures sticking out of different tissue types in Amphioxus. But the so-called golden age of cilia biology actually only started in the early 1990s, which is, for some of us, not that long ago. And so this actually all started with the discovery and description of IFT, and this was done in chlamydomonas. And this was the work of Keith Kosminski when he was a grad student in Joel Rosenbaum's lab at Yale. And this was a seminal paper that was published in PNAS in 1993. Following the discovery of IFT, the connection of cilia to ciliopathies really came to a head with the characterization of this ORPK mouse. And this is the Oak Ridge polycystic kidney mouse, which had been known for a long time, and it had also been known that the causal mutation in this mouse was this hypomorphic mutation in a gene, and this mutation was called TG737, and the gene was called Polaris. And nobody really knew what that gene did until working Chlamydomonas and C. elegans showed that the gene encodes a protein which is absolutely essential for IFT. And complete loss of function and mutations in that gene results in failure to form cilia. And so this then led to this whole, as I said, explosion of information about the role of cilia in these different ciliopathies. And so it is actually, I think that the study of cilia biology sort of provides an interesting case study for two important precepts in science. One is the role of serendipity. You sometimes find things that you weren't really looking for. So these early researchers who were found primary cilia weren't hunting for them. They were more interested in centriole and basal body biology. And the second part of this is that this also is a really great example of how unexpected findings in completely diverse range of model organisms, so Chlamydomonas, C. elegans, and even organisms such as Tetrahymena, Paramecium, Trypanosomes, have provided major lessons or major understanding of human diseases, of many human diseases. So our model organism of choice is C. elegans. And so C. elegans actually provides a lot of major advantages for studying the role of cilia in sensory biology. So first of all, these animals exhibit extremely complicated and complex experience-dependent sensory behaviors. And this is mediated by a very compact nervous system, of which only 60 of them are sensory. Secondly, unlike in the case of most metazoans, where most cell types are ciliated, in C. elegans, it's only these sensory neurons that are ciliated. And what that means in practice is that in the lab, you can actually isolate mutants, which are completely defective in cilia formation, but those mutants are perfectly viable and fertile. And then thirdly, the functions of many of these sensory neurons or individual sensory neurons are actually known. And so what that means in theory is that we should be able to correlate the structure of cilia of a given sensory neuron with its function. So in my talk today, I'm going to ignore most of the ciliated sensory neurons. I'm only going to be talking about a subset of these sensory neurons present in the bilateral amphid organs of the head. So there are essentially 2 of these organs, and they contain 12 neurons each. So let me just show you what the anatomy looks like. So this is the head of C. elegans, and what you're looking at is one of these sensory neurons called AWA. And this is the soma, the axon, and the dendrite that runs out to the nose where they ended these cilia structures. And these cilia, if you look at it head-on, are actually exposed to the environment through holes in the cuticle of the animal at the nose. And so what that means is that these cilia come into direct contact with chemicals, the stimuli in its environment. So as I said, the functions of many of these neurons are known, and this is painstaking work done over many years by many people. But the initial work done, I don't know what happened here. The initial work done on the identification of functions of some of these sensory neurons was actually done by my postdoc advisor, Corey Bergman, when she herself was a postdoc in Bob Horvitz's lab at MIT. And so here I'm just summarizing the known functions of each of these sensory neurons. And you can see they mediate attraction and avoidance to completely different sets of volatile chemicals, water-soluble chemicals, pheromone, temperature, and so on. And so just let me give you an example of the way we look at these behaviors in the lab. So a lot of the chemosensory behaviors we look at in the lab are actually done using these microfluidics-based devices. And so when I play this movie, what you're going to see is that we're running a stream of an aversive chemical through the center of this device. And you'll see that the worms will actually very assiduously avoid this chemical. And in fact, when they encounter the edge of this stream of chemical, they'll back up. And you'll also see as soon as we turn this chemical stream off, like now, they will now go back and no longer avoid the center. They'll move through the center. So this is actually a very rich behavior, and we can analyze the underlying components of this behavior and dissect them in great detail, and that's the subject of a completely different independent project in the lab. So what I'm showing you here is the response, aversive response to a specific pheromone, and that's mediated by this particular chemosensory neuron type. So of course, one of the reasons why these different sensory neurons have different functions is because they express different subsets of signaling molecules, such as chemosensory receptors. But another key contributor to the different sensory functions is actually the different ciliary structure. And this was first described in 2 seminal papers in the 70s and 80s by Liz Perkins and Sam Ward. And they used electron microscopy to describe the ciliamorphology of these different sensory neurons. And I think you can appreciate the diversity of the structures in this figure I took from the Ward paper. So you can see here some of these neurons have a single cilium, some have branched cilia, and then there's a whole bunch of other neurons which have extremely diverse morphologies and extremely complex morphologies. So, however, microscopy and imaging have come a long way since the 80s. And a few years ago, we came to the realization that if we wanted to obtain a higher resolution view of how these structures form and how they actually function, we needed to revisit their ultrastructure and their morphology using more modern imaging techniques. And so a couple of years ago, we set up a collaboration with a wonderful electron microscopist, Daniela Nicastro, who's also a GM-funded investigator. She was at Brandeis, and she very recently moved to UTSW. And the work that I'm going to talk about in the next couple of slides is the work of a former postdoc in the lab, in my lab, David Doroquez, and a postdoc in Danny's lab, Christina Birchu. And this work was also done in collaboration with James Anderson at University of Utah. So what they ended up doing was generating models, three-dimensional models, of the ciliated endings of 50 of the 60 ciliated sensory neurons in worms. So this was a fairly heroic effort, and so I just wanted to walk you through a couple of the steps that were involved. So what they did was they took worms, and then they cryo-immobilized them using high-pressure freezing, fixed them at low temperature using freeze substitution. They were embedded in resin, sectioned, and then imaged using transmission electron microscopy. And following this, they performed 3D reconstruction from serial section electron micrographs and also from serial section serial electron tomography. And then they generated 3D models of these sensory endings. Before I show you the models, though, I wanted to give you an idea of the resolution of the ultrastructure that they achieved. So this is a longitudinal cross-section through two different cilium types. This neuron is a member of the amphid organ that I've been referring to, and you can see the sort of different parts of the cilium. These are the microtubule bundles, and you can see at the base these microtubule bundles kind of flare out. This is a completely different cilium, not part of the amphid organ, and you can see this structure here, which is called a striated rootlet. And this is thought to play a role in maintaining ciliary integrity, and it's present in some, but not all, ciliated neurons. And that's also true in mammals. It's actually very strongly present, or it's a very prominent rootlet present in our photoreceptors and a subset of motile cilia, but much less prominently in other tissue types. So the quality of the ultrastructural preservation that they achieved is also visible in these cross sections, because I don't know if you can see this, but there's very beautiful conservation of the membrane bilayer and the characteristic organization of the 9 doublet microtubules that make up the axonium. So what they ended up doing then was collecting for every worm, they collected 170 70 nanometer sections. Each of these sections was then imaged, and this was about 140 image tiles. And so for every worm, they ended up obtaining 20,000 images. And they did this over 5 or 6 animals, just the nose. And so they ended up collecting 120,000 images. And then, of course, they had to go in and identify every single one of the cells and follow them across the sections. And they modeled, as I said, 50 of the 60 sensory ciliated endings. And together with the glia, this ends up being about 100 cells that they modeled. And since the C. elegans hermaphrodite has 959 cells, this was about 10% of all the worm cells. So in the movie that I'm going to play to you next, it's a 3D serial reconstruction model. And what you'll see here is that the colors, they've color-coded the different sensory neuron types. And what I'd really like you to appreciate here is the incredible diversity of the sensory endings. So if you concentrate on the yellow ones, the blue ones, the purple ones, you can see they look really different from each other. So this might be, actually, the diversity might be a little difficult to appreciate in this movie just because there's so many cells that are present here. So what I've done in the next slide is pulled out the models of 4 of these sensory neurons, and that's shown here. So these are the ciliated endings of 4 of the sensory neurons present in the amphid organs. So this neuron, we know its function. It's required for attraction to a subset of volatile chemicals. It has a cilium structure that defies description. We call it a Christmas tree, but I think it's actually far more beautiful than that. This is the AWC neuron, also mediates attraction to a different subset of volatile chemicals, also responds to temperature, completely different cilia structure. We call this a fan. And then these 2 neurons, ADL and AWB, both respond to repellents. to aqueous or volatile chemicals. They both have these branch structures, but that's essentially where their similarities end. They're, again, remarkably different from each other. And so the question here really becomes, how do these different types of structures form? And then, interestingly, why do they have to look like this? Why must they look like this for the functions of these neurons? What's the correlation? So I don't actually have answers to the second part of this question today, but we've actually made significant headway in understanding how these structures form, and that's what I'm going to talk about for the rest of my talk today. So one of the features that came out of this ultrastructural analysis was the location of the transition zone relative to the cilia branches. You might have noticed that a lot of the cilia I showed you had these branched morphologies. So in one type of branching, we noticed that the branches occurred at the transition zone. And looking at it in more detail, it turns out that these were actually not branches after all, but they were actually two independent ciliary axonemes templated by two basal bodies. In a second type, the branching occurs distal to the transition zone. So why is this interesting and or important? Well, I already told you that the transition zone plays a ciliary gatekeeper role. And so what this essentially means is that in these cilia, all the proteins that are required for the branching must be trafficked through the transition zone. In a third type of branching, the branching occurs proximal to the transition zone. And again, technically, this is actually not happening from the cilium. This actually occurs from a specialized dendritic tip. So we don't know how this localization is regulated, but what these data do tell us is that the trafficking mechanisms must be distinct from cilium to cilium. And this has turned out to be the case, and the first hint of this came from some very early work done in my lab by a former graduate student, Shoikat Mukhopadhyay, who I'm happy to say actually has continued on and looking at cilia biology, and he now has his own lab at UTSW where he's looking at ciliopathies and sonic hedgehog signaling. So I already told you that IFT is required to build these cilia, and it's the main transport mechanism within cilia. So one other advantage of C. elegans I did not refer to before is that you can actually watch IFT in vivo in single cilium types. And so this is a movie from Shoikat where this is again the nose of the animal, and these two are the rod-like cilia of these two sensory neuron types. And what you're looking at here is essentially the movement of a GFP-tagged IFT particle. And you can see very robust anterograde movement. You may or may not be able to see it. There's also good robust retrograde movement as well. And we can get a quantitative measurement of this movement over time in these chymographs. And so Shoikat asked whether this IFT mechanisms are actually used differently to build different types of cilia. So again, this is the same movie that I just showed you before, but here I'd like you to pay attention to the fact that IFT is occurring through the entire length of the cilium. So this is what the cilium looks like, the two of them, through the entire length of the cilium, although at different velocities, which we can calculate from the slopes of these lines. But something very different actually happens in this different cilium type, where I think you can clearly see that IFT is not as robust, and it's more or less restricted to sort of this proximal part of the cilium, and we see very little IFT, if any at all, in the distal parts. And so putting together some work that I don't have time to talk about today from my lab, and also primarily from John Scully's lab at UC Davis, what we now know is that IFT motors are indeed deployed differently to build different types of cilia. So for these simple rod-like cilia, John Scully's lab showed that two motors, the heterotrimeric kinesin 2 and the homodimeric KIF17 motor, work coordinately to build the middle, what we call the middle segment, and then the KIF17 motor works on its own to build the distal segment. But something very different happens in AWB. Here, these two motors also work together, but Shoikat found that KIF17 actually also has to move independently. And then we still don't know what exactly is building the distal part of this and why we see so little IFT going on there. That's something we're still trying to figure out. And then in the very different AWC cilium, the Scully lab showed that again, these two motors work together, but now they work together to build the entire ciliary axoneme. And so this idea of the deployment of IFT motors differentially to build different cilium types is actually now also turning out to be the case in many different mammalian cilia as well. So IFT not only transports structural proteins such as tubulin to build a cilium, it also plays a critical role in the transport of signaling molecules, such as transmembrane channels, receptors, and so on. And so given that IFT is being used differently in these different cilia, we were curious to know whether the mechanisms required for ciliary trafficking of these signaling molecules were also different among different cilium types. So in the next slide, I'm actually going to summarize the PhD thesis work of 2 graduate students in a single slide. So this represents about 10 person years of worth of work. It is, however, not all their entire thesis, just snippets of their work. So this is the work of Andrea Brier and Martin Wojtyniak. So this is a set of signaling molecules that are actually expressed in these neurons and are required for chemosensory signal transduction. So these include GPCRs and the cyclic GMP-gated channel subunits. And we also know that all of these proteins are, again, localized to these very different cilia. So Andrea and Martin both wanted to know whether these proteins are trafficked to these cilia via different mechanisms. And so Andrea actually looked at the trafficking and localization of a whole set of different GPCRs. I'm just going to show you one example of what she found. So here's a GPCR, and in the ASK cilium, it is actually localized very specifically through the entire length of the cilium. But in AWB neurons, if you express this same receptor in AWB, it is no longer present in the cilium. It's actually diffusely present throughout the entire cell, indicating that the mechanisms required for trafficking of this protein to the cilium or retention within the cilium are absent in this neuron type. Similarly, Martin looked at the localization of channel subunit, the cyclic GMP-gated channel, and he looked at this protein called TAX4. And again, in the AWB neuron, it is beautifully localized through the entire AWB cilium. But in ASK, it's also present in the cilium, but you can see it's only present just in this proximal region and completely excluded from the distal region. So that also tells us that the mechanisms required for ciliary trafficking of this channel subunit are distinct between these 2 cilium types and or the mechanisms that restrict the localization to a specific ciliary subcompartment. So Martin and Andrea went on to identify the trafficking mechanisms that are involved in the ciliary trafficking of these proteins. And again, I don't have time to show you this today. This work is published. They identified different trafficking mechanisms, but they also identified the same trafficking mechanisms that are used in different ways in these 2 different types of neurons. And so this is a mechanism by which you can also diversify the complement of proteins that are present in each of these cilia. So what I've told you so far then is our identification of cell-specific IFT mechanisms, identification of cell-specific mechanisms of ciliary signaling proteins, and I didn't have time to talk about this. We've also identified mechanisms that differ from neuron to neuron in the pathways that are required for trafficking ciliary proteins to the dendritic base. So I've been telling you the cilia are signaling organelles, and there were some very old data that suggested that cilia structures are not static. They're actually quite dynamic and can be further shaped and sculpted by sensory activity and also external cues. And some of the old evidence from this actually came from, again, work in Chlamydomonas. So in chlammy, when the 2 haploid gametes mate, the 2 flagella have to stick together. And when it sticks together, it activates a signaling pathway involving cyclic AMP. And in this figure from Ursula Gutnow's paper in 1980, you can see that this activation of the signaling pathway results in a very rapid restructuring of the ultrastructural as well as the morphology. So here's an unactivated and an activated flagellum. They look very different from each other, suggesting that sensory signaling can itself shape the cilia structure. And in the late 90s, Corey Barkman's lab also showed that loss of function in chemosensory signal transduction genes, such as this G protein, results again in very strong defects in cilia morphology. And so we were curious to know whether cilia shape can be further modulated by sensory activity, and whether this was something that was a feature of a subset of cilia, or whether this was something that was observed in many different cilium types in C. elegans. And so again, this is work done by Shoykath. So again, here is the list of signaling proteins that are expressed in and localized to the cilia of the AWB neurons. And what he found was that if he looked at the cilia structures in a subset of these signaling mutants, the cilia looked very different from each other. So here is animals that are mutant for guanylate cyclase, which is absolutely essential for the signal transduction in this neuron. And here's animals mutant for the cyclic-gated channel subunit gene. And in both cases, what you can see is that there has been a dramatic remodeling of the cilia shape. And this number here actually refers to the volume of this fan-like structure. So essentially, the membrane volume appears to change between wild type and in these mutants. So this suggests that sensory activity may actually play a role in shaping cilium structure. And he went on to show that this was actually very dynamic and regulated by external cues by doing the following experiment. So C. elegans in the lab, actually in the soil as well, eats bacteria. And what it responds to are chemical cues that are produced by bacteria. These are both volatile as well as aqueous cues. In the lab, you can actually grow C. elegans on a minimal media referred to as C. elegans maintenance media, or CEMM. So the worms will actually grow in it, but they're obviously no longer responding to any of the cues that bacteria will produce. And so Shoikat found that if you grew animals, wild-type animals, in this maintenance medium, this AWB cilia remodeled quite dramatically. And this remodeling it was very reminiscent of the kind of reshaping we observed in mutants that were defective in chemosensory signal transduction. But more tellingly, when he added bacteria back to the medium, he found that, again, the structure remodeled back to losing that sort of extra membrane volume, again indicating that these cilia are extremely dynamic in their structure and are constantly changing in response to external cues. And more recently, in collaboration with Oliver Black at University College in Dublin, we have shown that this remodeling in part is mediated by the altered balance of membrane delivery by exocytic mechanisms and membrane retrieval from the cilium by endocytic mechanisms. And what happens in sensory signaling mutants is that this membrane retrieval is slowed down, resulting in a buildup of membrane and reshaping of the structure. So to this list, then, we must also add sensory activity. And I should emphasize here that to date, we've only found that sensory activity plays a role in reshaping a subset of the cilia and not all. And this is something that I can talk about more at the end if anyone's interested. So the sensory-dependent or sensory activity-dependent shaping of structure is not something that's unique to cilia. It's been observed in many different structures in cells, and particularly it's been studied extensively in the context of synapses. And so analogous to what has been extensively described in synapses, we think that one reason for this is it's sort of a homeostatic compensatory pathway that tries to maintain cilium architecture and therefore cellular function in response to constantly changing environmental cues. This is a speculation, and this is something that we are still interested in, we are still pursuing, and it's an active area of ongoing research in my lab right now. Okay, so one mechanism that's actually currently missing from this slide is our identification of cell-specific structural mechanisms that may result in the generation of different types of cilium. And so in the last part of my talk, I'm going to tell you about some ongoing and as yet unpublished work on our identification of some of these mechanisms. And this is going to be, again, an interesting story of using C. elegans to find a completely new function for a protein that has previously been studied extensively in mammals, but not in the context of ciliogenesis, and we discovered this in C. elegans. So in order to address this, we actually decided to go back and take a look at the very first steps in ciliogenesis. So I told you the cilia are templated by the basal body. And so what that means is that wherever the basal body sits in a cell is going to determine where the cilium is going to form. And you can see these in these different types of mammalian cells. So in a polarized epithelial cell, the cilia are present on the apical surface, and the basal body is all dock at the apical surface. In non-polarized cells, the cilium is actually found close to the cell center. And that's because the basal body is actually positioned very close to the nucleus in the cell center. But in migrating neurons, the basal body actually is present in the leading edge of the cell, and again, that's where the cilium is present as well. And so obviously where the cilium is found in the cell and how it's oriented is absolutely critical for the cilium to be able to sense extracellular cues. And so it is also important then to make sure the basal body docks at the right place in the cell. However, there's almost nothing known about how basal body positioning is regulated. So this is also true in C. elegans. So I told you the cilia come out from the dendritic ends of sensory neurons, and that's where all the basal bodies are. So ciliogenesis, in the set of sensory neurons that I'm talking about, actually occurs in the embryo. And so what happens is these sensory neurons are born, and then their tips of their dendrites remain anchored in place, and then the cell body moves back to their final position in a process of retrograde extension. And so this was first described by Max Heyman and Shai Shaham. And so the basal bodies remain at the dendritic tips, and that's where ciliogenesis occurs. And I should say that the very first steps in ciliogenesis actually has yet to be visualized in C. elegans, the situation that we're trying to rectify right now. But in the same as in mammalian cells, nobody understands how the basal bodies end up sitting at the dendritic tips and how they remain sitting there. And so we reasoned that if we wanted to identify genes required for basal body positioning, then perhaps we could actually screen for mutants with defects in cilia structure, because defects in basal body positioning might also result in defects in ciliogenesis. And so the work that I'm going to be talking about next is the work of a former graduate student in the lab, Anik Olivier Mason, and has been carried out now by a really great postdoc, Ina Nechiporenko. So they identified mutations in this gene called GERDIN. And what they found, so here's the AWB cilium, again, here's in wild-type animals, and they found in GERDIN mutants that you often had these cilia defects. Instead of the 2 branches or 2 axonemes, you saw 1. And more tellingly, something you never see in wild-type animals, you see this dendritic shortening, which suggests that the dendrite is no longer staying anchored in place, but is actually being truncated. And this can be complemented by a wild-type copy of the gene. And so this is a quantification of the defects we see. I should point out this is a hypomorphic mutation. Null mutations in this gene result in lethality. So what they also found is that the defects that they're observing here, they observe in a subset of ciliated sensory neurons and not all. Again, indicating that whatever this gene is doing is acting differently. It's doing this differently in different types of ciliated sensory neurons. So they cloned this gene. And so GERDIN is a member of this incredibly conserved GERDIN protein family. And so this is a large coil protein with a domain at its N-terminus, which is called a hook domain, predicted to bind microtubules. Has a much more divergent C-terminus, which depending upon the species has a G-alpha binding motif, can have a GEF motif, can bind actin, also has an AKT phosphorylation site. So this protein actually has been studied quite extensively in mammals in the context of cell migration. In metastasis, so this appears to be a great marker for invasive tumors. It's highly upregulated in invasive tumors. It has been studied extensively in the context of neural migration during development, also wound healing. In all of these cases, we require cell migration. It's also been shown to be present in centrosomes and play a role in cell division. And then in flies, there are 2 very recent papers which suggest that it plays a role in anchoring or linking the lesion complex to the actin cytoskeleton. And there's a very recent paper which suggests it plays a role in regulating morphology of dendrites in a subset of mechanosensory neurons. And so taken together, this seemed like a really good candidate for a protein that might link the basal body to the cytoskeleton and keep it in place. So Ina asked whether the ciliary defects we observed in C. elegans were actually correlated with defects in basal body positioning. So she looked at the localization of basal body proteins. So if you look at this particular neuron here, this is a basal body protein that is localized right to the base of the cilium, which is where it's always found in wild-type animals. But in GERDIN mutants, what she finds is that this particular protein ends up sitting in some random ectopic location in the dendrite. Sometimes it's even in the cilium, and sometimes, very rarely, it's more diffuse. And that's shown here in cartoon form and quantified here. This is something you never observe in wild-type animals. And moreover, when she looked at the localization of different basal body and transition zone proteins, she found that all of them were co-localized together in these ectopic locations, in these ectopic puncti, suggesting then that the basal body and transition zone is assembled correctly, but it doesn't sit at the right spot. It moves around and is misplaced in GERDIN mutants. And so we corroborated this with ultrastructural analysis. This is done with our collaborators, Ollie Black at University College. And so in wild-type animals, you see this transition zone right at the base of the cilium, and in Gurdon mutants, you can see this fully assembled transition zone in sort of much more proximally in the dendrite. And so this number here refers to this section here, starting at the nose. So sometimes this transition zone is kind of freely floating in the cytoplasm, and sometimes it docks to the membrane, but in the wrong spot. And the transition zone has a very unique ultrastructure. And so we can see that the zone is actually assembled correctly, but again, not sitting at the right spot. So where is Gurdon present? Well, it localizes right at the basal body. So this is co-localization with basal body proteins. And what she finds is that it actually co-localizes with the basal body protein, but appears to be enriched to the proximal domain of the basal body. And it's actually completely excluded from the transition zone, which is sort of more distal to the basal body. So what this indicates is that Gurdon is a protein that plays a role in anchoring the basal body in place at the base of the cilium, and that in Gurdon mutants, you see sometimes partial mislocalization of the basal body, resulting in cilia defects, and sometimes there's complete mislocalization, resulting also in dendritic shortening. So I already told you that Gurdon has been studied a lot. It's been studied in the context of cell migration issues, but has never been actually implicated in ciliogenesis in mammalian cells. And so given what we just found, that it plays a role in basal body positioning and ciliogenesis in worms, we wanted to know if its role in ciliogenesis is actually conserved in mammalian cells. And so we looked at this particular cell line, human retinal pigment epithelial cells, which actually forms cilia very robustly when you pull them out of the cell cycle. And so this is control cells, and almost 100% of them form cilia, and you can see that here by staining with different ciliary antibodies. But when Ina knocks down Gurdon, she sees a very dramatic defects in ciliogenesis. And what we also found is that just like what we saw in worms, these ciliary proteins are all actually recruited to one spot, but there's no productive ciliogenesis in this case. And even the cilia that do form are actually significantly shorter in length. Are there defects in basal body positioning? So this is a non-polarized cell line, and the basal body stained here with antibodies against a centriolar protein is very closely located to the nucleus, stained here with DAPI. And upon knockdown of Gurdon with siRNA, you can see that the position of the basal body with respect to the nucleus becomes far more variable. And we often see positioning at distances greater than 4 microns from the nucleus, something you never observe again in control animals. So again, we've seen parallels between what we observed first in worms. We see the same thing in these human cells. And so does Gurdon also sit at the basal body? And indeed it does. So this is staining with antibodies against Gurdon, as well as markers for the cilium. And you can see here very clearly that it localizes just to the proximal parts of the centrioles, exactly what we saw in C. elegans. So the proximal domain of centrioles is sort of an interesting region, and it contains another protein called CNAP1 or CEP250, centriolar protein 250. And consistent with Gurdon localization in these same domains, we can see perfect co-localization with CNAP1. And Ina went on to show that this protein CNAP1 is actually required to recruit Gurdon, because if she knocks down CNAP1, you can see the amount of Gurdon that then get recruited to the proximal centrioles is not gone, but it's significantly diminished. So why am I talking about CNAP1? Well, CNAP1 actually has been studied extensively in the context of recruiting another very important protein called rootletin. So rootletin is a component of this proteinaceous linker that links the two centrioles together, and then this linker is dissolved during mitosis. But another component, another role that rootletin has is it's component of these ciliary rootlets that come out of the proximal domains of these centrioles and essentially anchor the basal body to the actin cytoskeleton. And previous work, both in mammalian cells as well as in C. elegans, had shown that rootletin may actually have a role in ciliogenesis, and there were defects in ciliogenesis in both of these systems. And so we asked the question of if CNAP1 recruits Gurdon, and if it's also known to recruit rootletin, is it actually recruiting rootletin in part by recruitment of Gurdon? In other words, if we knock down Gurdon, would we actually see defects in these rootletin structures or rootletin recruitment? And so that's what Ina did here. So again, this is rootletin present at the centrioles. And you can see upon Gurdon knockdown that the amount of root lectin that is present at the centrioles is actually significantly diminished. So far we've actually used worm work to inform our mammalian work, but now we're going to go in the other direction. We actually now found a mechanism for Gurdon function in mammalian cells, and we now ask the question, can we use that information to go back to worms and see if it's the same mechanism in C. elegans? And so the homologue of root lectin in C. elegans is this protein called Ki10, and normally it's present in a very tight set of puncta right at the base of cilium. And just like what we saw in mammalian cells, in the Gurdon mutant, we found that there was decreased root lectin, and also the root lectin that was present was actually mislocalized. It was present in ectopic locations, again, throughout the dendrite. So the model that I'm building so far is, or I've built so far, is that what Gurdon is doing is essentially anchoring the basal body in place via root lectin. And so if that were the case, then you would expect that knocking down root lectin should have the same phenotypes as Gurdon knockdown in terms of both basal body positioning and cilia defects. And Ina also found this. So if she knocks down root lectin, again, you see basal body mispositioning, the same phenotype we observed when we knocked down Gurdon. And she also observes strong defects in ciliogenesis. So putting all this together then, we have very similar mechanisms of functions of, new functions of a well-studied protein that we uncovered first in C. elegans, but appears to do the same thing in human cells, in these mammalian cells. In both cases, Gurdon is found at the basal body, in the proximal basal body. In both cases, it recruits root lectin. And in both cases, if you knock down Gurdon, you see defects in basal body positioning and also ciliogenesis. So one thing that's actually kind of interesting here is that I told you that Gurdon has been implicated in cell migration, for neural migration, wound healing, which also requires migration. And cell migration actually has been linked to basal body positioning and cilia. And given this new role of Gurdon that we have now uncovered for it in basal body positioning and ciliogenesis, we want to now know whether this role of it in basal body positioning underlies, either wholly or in part, its previously described roles in migration, in cellular migration. So that is something we're looking into right now. So tying this back, however, to the major theme of my talk, we also noticed that the defects of Gurdon are only observed in a subset of C. elegans sensory neurons. And so Gurdon falls firmly into the category of structural proteins that are, again, used differently in different cells to build different types of cilium. So to summarize, what I've told you today is our work in identifying the mechanisms, both hardwired as well as plastic, that work together to generate different types and these unique cilia architectures. So we're continuing to identify and characterize these mechanisms, and we're very interested especially in the intersection between these two pathways. But a major unsolved puzzle that we're looking forward to tackling in the future is really this sort of unanswered question of how the forms of these cilia dictate function, and in turn, how the function feeds back to further modulate shape. And so this is going to be the subject of much work in upcoming years, and I hope that we'll have data to share on this in the future. So I'll end there, acknowledge the people who did the work. So the group of people who work on cilia biology in my lab have named themselves the Cilia Squad. And so this is in blue are the people whose work I talked about today. So these are all people who have since left the lab. Martin is actually at the NIH. And I talked about the current work, the work of a current postdoc in the lab, Ina. And other people here are also doing very exciting work that I did not have time to talk about today. So everybody says this, but it's really true. One of the fun things about doing science is the opportunity to work with a lot of very smart, very interesting people. And I've had the real good fortune of working with a bunch of very wonderful collaborators. So one of them being Danny Nicastro, who we collaborated with on the EM work, and we're continuing to collaborate with. Ollie Black, who is a longtime collaborator on our C. elegans work. Max Heyman, who we've been recently collaborating with on the Gurdon work. And all the cilia work in my lab is currently funded by GM. It's been funded in the past also by DCD. I also have completely independent projects in the lab working at the neural mechanisms of chemo and thermosensation, which are currently funded by NSF and also by GM. So I'll end there, and I thank you for your attention. I'm happy to take questions.
Thank you very much for a great talk. I'm intrigued by the structures that you showed. Are there any adhesion molecules or any things that anchor the membrane in different shapes in the different neurons?
So are you talking about the cilia or the tips of the dendrites? Talking about the cilia?
The cilia.
Yeah, so so we looked at that very carefully, and it seemed like this would be a good place for there to be adhesion complexes, like it's been shown by Jennifer Lippincott-Schwartz's lab. And so we, because they're actually all, they're all sort of in this channel, and they're all sterically restrained within that channel. So they should be localized in place. And one thing I didn't mention is that also when you look at the two branches of different cilia, they're always in a very characteristic location with other cilia in between. But we haven't so far seen any evidence of any kind of adhesions at the cilia. There are some at the base, but not among the cilia themselves.
I think what I was trying to ask is between the cilia and other cells around that would pull them into these shapes.
Yeah, so that's right. So that's the glial support cells that are around them. And so far in the cilia axoneme structure themselves, there's nothing holding them in place there. There are structures at the base.
Thanks.
So there are junctions at the base that hold them in place.
Well, congratulations for revealing some of the features of the cilia biology. So in some of the structures you have shown, is not continuous, they are punctates. In the ASK model of the cilia, they are not continuous. So what are the causes of these fragmented segments?
The axoneme structures themselves are contiguous. So are you referring to the IFT?
Yeah, well, actually you showed some structures of the cilia where they are not continuous. These are the fluorescent microscopic imaging. So I was wondering, what are these gaps?
Oh, I see, yeah. So those are actually different ciliary subdomains, and some of these proteins are very specifically restricted to particular cilia subdomains and excluded from others. So what you're looking at for sometimes you'll see is cilia proteins present in the axoneme part, but completely excluded from the transition zone. And that's why you end up seeing a gap, and then it's present again at the basal body. And so there are all these different ciliary subcompartments, and some of these proteins are just present in a few of them and not in others, which ends up looking like there are gaps, but they are actually contiguous structures.
So what is the role of your Guardian-1 in mammalian system?
So the idea is that it's actually playing a role, at least in this particular context, and again holding the basal body in place and allowing it to dock to the membrane or connecting it also to the cytoskeleton, and so that it ensures that it's in the right place for the cilia to form at the right spot. That's the general idea.
Great work, thank you.
You have shown that there are different IFT motors that work differently to generate different cilia. Is there any evidence to support the possibility that the gating proteins work differently to generate different cilia?
Absolutely. So the transition zone that I referred to is actually a complex of a very large number of different proteins. Some of those proteins are actually expressed differentially among different different neuron types or different cell types. It's actually not just work in C. elegans. There's a lot of work done on this in mammalian cells. And again, some of them are actually present at different locations and used differently in different types of cilium. So definitely the gating properties are different. One thing I didn't refer to also is, in addition to the transition zone, there's also what looks like a nuclear pore complex at the base as well. So you see proteins, this is work from Kristen Verhey at University of Michigan. You see proteins like nucleoporins, they're important, so those are also present in the cilia base, as well as proteins like septin. So it's an incredibly complex gate structure and used very differently among different cilium types to allow specific proteins to go in or stay in.
Okay, please thank Piali one more time and join us in the library for refreshments.